Hello, everybody. Thank you to be with us today. I'm Carol Tonello. I'm in charge of business development and applications in Hyperbaric. And I will now briefly present you our innovation in the HPP Innovation Week, which are the HPP Research Awards. So just let me share my screen. The objective of the HPP Research Award is the dissemination of the latest scientific advances on high-pressure processing. And what we really would like to succeed is to share with you the latest innovation we have in the lab, coming from young scientists. We had 30 applications, and the deadline was May 23. And during the HPP Innovation Week, we will have the pleasure to see the 3 finalists of each category. So the category are human and pet nutrition. So we think that it's very important for the development of HPP in the industry and in the world in general, to take care about the sensory and nutritional quality of the product, the bioavailability, the stability during the storage, the sensorial quality, and the valorization of the product post extraction. The second category we have for these awards is the food safety and the shelf life extension. The main application of HPP is in activating microorganisms, pathogen, and spoilage. So this is very important to see what are the latest innovation, what are also the benefits and the limits, and so the finalists we have selected give from our sense, give the most interesting last advances in this category. And the third one, which is very important also today, is the sustainability and the product development. HPP is used for new products which need to be sustainable, and this need to include the life cycle assessment, for example, and also a good, interesting product development. The jury, we wanted to have different profiles, and the member of the jury, the selection was made by from the 30 candidates. The selection was made by Hyperbaric HPP application team, and the finalists will be selected with the jury you can see here. One person, Samantha Escobedo, she's a professor at the University Tecnológico de Monterrey in Mexico. Chris Duna is a senior scientist and director at the Natic Soldier R&D Center. So he is a civilian belonging to US Army. And also, I'm a part of the jury representing the industry, and having an experience of about 30 years in HPP. The requirements, the participants are graduate or intergraduate student, postdoctoral researcher, young professional with a graduation date between 2018 and 2022, because we wanted really to have young scientists, also to give them an exposure to all the people participating, watching the Innovation Week. The research topic nutrition, food safety, and sustainability. And the type of publication accepted are peer-reviewed scientific journal publications, oral communication, or poster communication presented in congresses. So we will have the presentation of the three finalists of the three categories, and the last day, after the last session, we will have a HPP Research Award winner ceremony with the jury announcing the results. And I hope you will enjoy these HPP Research Awards. Thank you very much. So now I will give the floor to my colleague, to Dr. Rui Queiroz, and he will have the pleasure to present you the first finalist of the human and pet nutrition.
Hello, everyone, and welcome to the first edition of the HPP Research Awards. My name is Rui Queiroz. I am part of the applications team at Hyperbaric, and today I will be presenting the three finalists from the category Human and Pet Nutrition. Our first finalist is Prakar Chatur. Prakar is a researcher with expertise in product development involving grains and legumes and novel technologies. He holds a PhD from the University of Curtin, where he worked with HPP and chickpeas. And today we will be presenting his work titled The Effect of HPP on Textural, Bioactive and Digestibility Properties of Cooked Chickpeas. Prakar, you can share your screen now and start your presentation. Best of luck.
Thanks, Ru. Hi, everyone. So in the HPP Innovation Week, the title of my presentation is The Effect of High Pressure Processing on Textural, Bioactive, and Digestibility Properties of Cooked Kimberley Large Chickpeas. Now, why chickpeas or why high pressure processing? Australia is a major producer of chickpeas in the world. Chickpeas are a rich source of plant proteins, dietary fiber, and minerals. However, there is a lack of information on effects of HPP on legumes, specifically chickpeas. Effect of HPP on lentil, mung bean, and pea starches are available. However, there was no information on digestibility and antioxidant properties of chickpeas. HPP could be used to produce a ready-to-eat convenient chickpea product with increased shelf life and no adverse effects to the nutritional properties. Kimberley large chickpeas grown only in the Western Australia were used in this study. The hypothesis behind this study was high-pressure processing will not change the nutritional properties of cooked Kimberley large Kabuli chickpeas, and the experimental design comprised of 3 pressure settings, which were 200, 400, and 600 megapascal, and 2 treatment times, which were 1 and 5 minutes, giving a total of 6 processing combinations. The processing steps included soaking of chickpeas for 12 hours in excess water, cooking in boiling water for 30 minutes, cooling under running tap water, removing excess moisture, and vacuum packaging followed by HPP processing. Cooked untreated samples were used as control. The assays which were performed on these samples, including the control, were texture profile analysis, including firmness, cohesiveness, springiness, gumminess, and chewiness, nutritional properties, proximate composition, including dietary fiber, starch content, and minerals, bioactive properties, including total polyphenol content, DPPH antioxidant capacity, ABTS, and ORAC antioxidant capacity, as well as digestibility assays, including in vitro protein digestibility and in vitro starch digestibility. Looking at the results, we can clearly see that the firmness values for HPP-treated samples were significantly lower than the control, and the cohesiveness, gumminess, and chewiness values as well were lower than the control, which resulted in a softer texture of the HPP-treated samples, specifically the samples treated at 600 megapascal for 5 minutes. Looking at the nutritional properties, there were no significant differences in the nutritional composition of samples, including the control, except for the starch content, which was significantly lower for samples treated at 400 and 600 megapascal, and the soluble dietary fiber content of sample treated at 600 megapascal for 5 minutes was significantly higher than all of the rest of the samples. The starch reduction can possibly due to leaching of starch while processing was occurring, and thus HPP can be used to reduce starch of high starch grains and pulses in the future. Looking at the bioactive properties, the total polyphenol content of samples treated at 600 megapascal were significantly lower than all of the other samples, including the control. The possible reason behind it could be the enzymatic oxidation of samples which occurred during processing. And the DPPH and ABTS antioxidant capacities were also determined. And from the figure on the right-hand side, we can see that the DPPH antioxidant capacity of samples treated at 400 and 600 megapascal was significantly lower than all the other sample, whereas the ABTS antioxidant capacity of sample treated at the highest treatment levels was the lowest. Similarly, the oxygen radical antioxidant capacity of sample treated at the highest level, which was 600 megapascal for 5 minutes, was the lowest and was significantly different than all other samples. The reduced antioxidant activity may have occurred due to cooking and high pressure processing, as well as destruction of bioactive components in the chickpeas. Coming on to the scanning electron microscopy of isolated starch from freeze-dried samples, the first figure represents raw starch granules from raw chickpeas. The second figure, which is B, represents starch granules from cooked chickpeas, which was the control, and figures C to H represent starch granules of high-pressure processed samples. It was evident from the scanning electron microscopy that high pressure induced damage or changes to the starch granule. We can see there were fissures or damages on the starch granules, specifically for samples treated at 600 megapascal, which might have occurred due to changes in the amylopectin chains, their interaction, as well as internal changes which might have occurred due to high pressure processing. Followed by scanning electron microscopy, we also performed Fourier transform infrared spectroscopy, which clearly showed changes in the spectral region related to starch intermolecular structure, specifically at 1140, 1040, and 1020 wave numbers. From the figures, we can see that there were some negative spectra decline in both figures, but the intensity of decline of spectra in the figure for samples treated for 5 minutes at 3 different pressures was intensified. Followed by Fourier transform infrared spectroscopy, I performed starch digestibility assay, which showed that samples treated at 200 megapascal for 5 minutes and 600 megapascal for 1 and 5 minutes both showed increased slowly digestible starch content and reduced resistant starch content. Slowly digestible starch is the most desirable starch and leads to slow release of glucose in bloodstream after a meal is consumed, and thus can lead to a low glycemic response in human body. Coming on to the in vitro protein digestibility and mineral content, there were no significant differences between the control and the highest treatment levels in the in vitro protein digestibility and mineral content as well. However, the mineral content was slightly reduced in the high pressure treated samples. So the main findings which I found after conducting this experiment were change in the texture profile of chickpeas with an increase in pressure during high pressure processing, no significant changes in the proximate composition and mineral content of samples. These findings were published in a journal article in Frontiers in Nutrition, and the novel findings from this experiment were changes in the spectral region related to star structure and in the morphology of isolated starch molecules, and changes in the digestibility of available starch, as well as a decline in the polyphenol content and antioxidant capacity of high-pressure processed chickpeas. Future perspectives include understanding the mechanism behind changes in these nutritional composition of other foods due to high-pressure processing, new product development using high-pressure processing for value addition, as well as exploring the potential of chickpeas in the functional food market. I would like to thank my supervisors as well as Curtin University for letting me pursue my doctoral degree and on a full scholarship. Thank you so much.
Thank you for the presentation. It was very clear and it was a nice presentation. I do have a couple of questions for you now. The first one is, what were your main difficulties working with HPP and ready-to-eat chickpeas?
So the biggest difficulty I personally had was getting my samples processed, because the processing facility was around 3,000 kilometers from my location. So the samples had to be sent, as well as, they had to come back. And because I wanted the samples to hold their integrity, they were shipped overnight by a freight, by airplane. And then after processing, we received them within, I would say, within 10 hours in a secured, fully packaged freezer box. So that was one of the biggest, I think, challenges for me to basically manage all of this.
Yeah, the difficulties of doing research in a very large country.
Yeah.
And the next question is, based on your research, what are the main advantages of using HPP to process chickpeas and other legumes?
So in my experience, one of the biggest advantages was the increase in slowly digestible starch levels. And as I mentioned, slowly digestible starch is the most desirable form of starch in any food, which leads to slow release of glucose in the bloodstream after a meal is consumed. And it basically leads to a low glycemic response. So it can be considered as a health benefit of consuming slowly digestible starch. Yeah.
Yeah, that's great, and a new application for HPP. Well, that's all. Thank you for participating, and best of luck with the awards.
Thank you.
Continuing with the presentations from the finalists of the HPP Research Awards, we now have the presentation of Cristina Serra. Cristina is doing her PhD in the Food Safety and Functionality program at IRTA in Spain, where she evaluates the application of HPP to control pathogenic bacteria in meat products. Her presentation is titled Development of a Tool to Evaluate the Efficacy of the HPP to Guarantee Food Safety of Acidulated Raw Pet Food. Cristina, you now have the floor. You can share your screen, and good luck.
Rui, can you confirm me that the screen is seen okay?
Yeah, everything okay. Can go ahead.
Thank you, Rui, for this nice introduction. I would like also to thank all the committee for giving us the opportunity to present our recent work in raw pet food. The consumption in raw pet food is increasing, and this is for that raw pet food diets are perceived as more nutritious and natural, as the components are not heated and maintain the thermosensitive component, which is thought to give potential benefits, including, for example, improved behavior or shiny coat, among others. However, this lack of heating treatment as a key step can favor or can lead to a health risk. And this is because raw pet food can harbor some pathogenic bacteria like Salmonella, Listeria monocytogenes, or Escherichia coli. For example, in case of Salmonella, and due to its low infection dose, there are some regulations what a zero tolerance is imposed for some raw pet food producers. For that, raw pet food producers must look for strategies to minimize this possible risk and also to accomplish with legislation. And one of these strategies can be high-pressure processing, which is a non-thermal technology applied to kill pathogenic bacteria while maintaining the nutritional and freshness traits. However, when we applied high-pressure processing, we must take in consideration that its efficiency depends on several factors, including, for example, technological factors such as the pressure, the time of pressurization, or the temperature of the fluid, as well as the microorganism we want to inactivate or the food matrix. In this framework, we have to keep in mind that the microbial response to high-pressure processing is variable, being a species and strain dependent, and also that the pressure resistance of microorganisms depends on the food matrix characteristics. For this, the aims of the present study was to, on one hand, to determine the pressure resistance of different strains of Salmonella, Listeria monocytogenes, and Escherichia coli in raw pet food, and the second aim was to study the inactivation kinetics of the most pressure-resistant Salmonella strains in raw pet food. For the first aim, we included several strains of each pathogen in the study. These strains had different serotype and belongs to different origin. Also, for each pathogen, Salmonella, Listeria, and Escherichia coli, we include types of strain. And in case of Listeria monocytogenes, we also include some strains from the European Reference Laboratory, the ones that are recommended to be used in challenge tests involving meat products. What we did was to take two different kinds of raw pet food products, one that we named control, and another one where lactic acid was added to obtain an acidulated product. Here you can see that the acidulated had a lower pH compared to the control product. These products were inoculated with the different strains of Salmonella, Listeria monocytogenes, and Escherichia coli at 10 to 8 CFU per gram. Samples were vacuum packaged and when pressurized at 400 megapascals for 5 minutes. Immediately after pressurization, the pathogens were enumerated in case of Salmonella on chromogenic agar, in case of Listeria monocytogenes on chromogenic Listeria, and in case of Escherichia coli on Rebecca. And then the log reduction, the difference between the amount inoculated and the enumerated after high pressure processing was calculated. For the second aim, both kinds of products were used, the same that we used in the for the first aim. But in this case, we inoculate the three most resistant Salmonella strains at 10 to 8 CFU per gram. Samples were also vacuum packaged, but in this case, pressurization was carried out at 600 megapascals from 0 to 10 minutes. Immediately post-high pressure processing, the amount of Salmonella was also enumerated on chromogenic Salmonella agar. But also in this case, and also to assess the potential recovery of Salmonella strains after high pressure processing, half of the samples were kept for 24 hours at 4 degrees before the enumeration. Results were modeled, and in this case, the log logistic width tape model was applied. This model gives us two different kinetic parameters, including Kmax, which is the inactivation rate, and T-shift, which is the time for the appearance of a resistant tail. Regarding the results obtained from the first aim, here we see that one graph corresponds to one different microorganism, Salmonella, Listeria monocytogenes, and E. coli. Here we have the pressure resistance of the different strains assessed. In red, we see the pressure resistance of the strains in control product, and in green, we observe the pressure resistance of the strains in acidulated products. We can see that in the majority of cases, the inactivation of the strains in acidulated products is higher compared to control. However, there is a lot of variability between strains of the same microorganisms. In this framework, we can see that, for example, if we focus on the type strains, we can see that for Salmonella and Listeria monocytogenes, they are very susceptible to high pressure processing, but in case of Escherichia coli, the type of strain was one of the most resistant. If we focus on Listeria monocytogenes and in the strains that belong to the European reference laboratory, the ones that were recommended for using in challenge tests, we can see that also we can find a lot of variability between them. Especially there are ones that are susceptible to high pressure processing and will be not recommended to be used in validation processes. In case of Salmonella, we can identify 3 strains that were so resistant to high pressure processing. And for this, these strains were selected to perform the kinetic analysis. If we focus on the kinetic analysis performed, that was the second aim of the present study, here we can see that each graph corresponds to one different Salmonella strain, CTC 1022, GN0082, and GN0085. In red, we can see the inactivation kinetics of Salmonella in control products immediately post high pressure processing, and in green, we can see the inactivation of Salmonella in acidulated products immediately post high pressure processing. We can see that the inactivation of Salmonella is higher compared to control, and especially this Salmonella, although is increasing along the pressure holding time, we can find that there are some restrictions. If we look on the Chemex, we can see the inactivation rate of Salmonella is systematically higher in acidulated products compared to control. However, the other parameter that gives us the log logistic with tail model, the T-shift, we can see the appearance of a resistance tail. What does it mean from the technological point of view, the appearance of this resistant tail? It means that whenever you increase the pressure holding time, no longer inactivation of Salmonella will be increased. So this is a technological parameter important to be taken in account when defining the process parameters of our own process. If we don't only focus on the inactivation of Salmonella only when we enumerate it immediately post high pressure processing, we can also observe difference with these counts made 24 hours post high pressure processing. All the inactivation curve move upwards, especially those from control product. We can see that these 24 hours results in a lower inactivation of Salmonella. What does it mean? What does it mean that during these 24 hours at 4 degrees, Salmonella was able to recover some viability, and for this we obtained lower inactivation. However, this was not a clear case for the acidulated samples, the ones that lactic acid were added. And here you can see that if we look at the samples that were enumerated 24 hours post-high pressure processing that were acidulated, that no statistical difference were observed between those obtained immediately post-high pressure processing. Let's focus now on, as we observe that all the strains have the same behavior regarding the impact of these 24 hours post-high pressure processing, we can focus on now only on CTC 1022. We observed that enumerating the counts of Salmonella 24 hours post-high pressure processing results also in a lower inactivation rate compared to that obtained when enumeration of Salmonella was carried out immediately post-high pressure processing. And these kinetic parameters obtained can be used as a tool. And a tool for what? And a tool to estimate the holding time, this pressure holding time needed to inactivate this 5-log of Salmonella. If we focus on the results obtained immediately post-high pressure processing, we can estimate that a minimum of 5 minutes will be needed to inactivate 5-log of Salmonella in control products. This time will be of 3 minutes for acidulated products. However, if we look at the results obtained 24 hours post-high pressure processing, this time will be doubled, raising at nearly 10 minutes for control products. And in case of acidulated, this increment time will be lower, but still will be higher than the one estimated immediately post-high pressure processing. This, what it means is that if we estimate, we validate our processes only considering the results obtained immediately post-high pressure processing, we can overestimate the effect of high pressure processing. Thus, the conclusion of this study was that in raw pet food, high pressure processing inactivation of Salmonella, Listeria monocytogenes, and Escherichia coli strains shows wide variability. Identified high-pressure processing resistance strains can be used in challenge tests to assess and validate the efficacy of high-pressure processing. Moreover, we here put in front the relevance of sublethal injury in the overestimation of the immediate effect of high-pressure processing. Also, that acidulation enhances high-pressure processing lethality and minimizes recovery of sublethal damage in a species and strain-dependent manner. And also that here we develop a tool to evaluate the efficacy of 600 megapascals treatments and allows to set the pressure holding time required to achieve the target lethality of Salmonella in raw pet food. All these results and conclusions can be found in our recent paper, Enhanced High Hydrostatic Pressure Lethality in Acidulated Raw Pet Food Formulations, was pathogen and species and strain dependent. However, besides all these results presented until now, I would like also to focus on the impact of these results. Here, we have characterized a versatile pool of Salmonella, Listeria monocytogenes, and Escherichia coli strains available at IRTA for validating the efficacy of high-pressure processing in meat products with similar physicochemical characteristics to raw pet food. Also, this study presented here today was the base to develop a decision support system to optimize the formulation with lactic acid and high-pressure processing treatment. And the results of this decision support system developed has been recently published the last week in a paper titled High-Pressure Processing Inactivation of Salmonella in Raw Pet Food for Dog is Enhanced by Acidulation with Lactic Acid. Finally, I would like to thank to all the people being group of the Food Safety and Functionality Program of IRTA, and just mention that this work is part of my PhD thesis, and in this framework, just to say a thank you to my directors, my PhD directors, Anna Joffre and Sarah Bebefsit. And I would like to finish this presentation just saying thank you for being here, and if you have any questions, I will be glad to answer. Thank you.
Thank you for the nice presentation, Christina. It was very clear and it's a very interesting study. I do have a couple of questions for you. The first one is, what were your main challenges or difficulties working with HPP and raw pet food?
The main challenges in this case was to optimize the protocol to prepare the strains before even Listeria, Salmonella, or E. coli strains to be used in these challenges, because it's really important to have the same physiological state of the strains when we want to compare the different strains resistance of the different microorganisms. For this is really important how do you prepare the strains, and also the time that takes to you to prepare the strains.
Okay, makes sense. And the next one, do you think that adding lactic acid to the pet food formulation has any limitations or problems?
It enhances the lethality of pathogens with high pressure processing, but also we have to keep in mind that lactic acid it's really tricky. You have to play, let's say that you have to play with the amount of lactic acid you add in the raw pet food, not only when you focus to inactivate the bacteria, but also you have to keep in mind that this raw pet food has to be consumed for the dogs or the cats, so it don't have to affect the palatability and the taste of the product, no? And in this case, what we did is to play with these amounts of lactate that didn't affect the quality of the product, and the pets didn't notice that lactic acid was there.
Okay, very nice answer. Thank you for your participation and great presentation in these HPP Research Awards, and best of luck.
Thank you very much.
The next presentation from the finalists of the HPP Research Awards in the category of Human and Pet Nutrition is from Nupur Gozavi. Nupur has a PhD in food science from Rutgers University, where she worked with HPP to enhance the nutritional properties of fruits and vegetables. She is currently an innovation scientist with Beyond Meat, and she will be presenting her work titled High Pressure Assisted Infusion of Calcium in Fruit and Vegetables. Nupur, you can share your screen now and start your presentation. Good luck.
Thank you, Ray. All right. Hello, everyone. My name is Nupur Gosavi, and I'm currently a scientist at Beyond Meat, and I'm delighted to present my PhD research that focused on high-pressure assisted infusion of calcium in fruits and vegetables. I'm also excited and honored to be a finalist in the Human and Pet Nutrition category at HPP's Innovation Week. So let's just dive straight into the talk. My talk is divided into 3 sections, and we'll start with the introduction. As everyone here knows, high-pressure processing is a non-thermal batch processing technology that subjects foods to extremely high pressures that leads to improved food safety, improved food quality, and also extension of shelf life. We also know that when you subject solid foods like fruits and vegetables to high-pressure processing, there are textural changes due to cell breakage that can occur. So what we thought when we started this PhD research was, can we go beyond these traditional applications of high-pressure processing and leverage those textural changes and the cell breakage to our benefit and utilize it to infuse materials? As it so happens that there were a few research already existing that looked at infusing some nutrient molecules into different foods. So one of these researchers actually came from our lab, where we looked into infusion of quercetin, which is an anthocyanin, into cranberries. And if you look at this graph here, you can see that the amount of quercetin that was infused in the osmotic infusion was actually 10 times lesser than the one infused in high pressure processing within 10 minutes. That means we got 10 times higher infusion in HPP within 10 minutes, and this infusion was almost enhanced and accelerated because it took 24 hours for osmotic infusion to reach the same level. So now that we know that HPP can enhance and accelerate the mass transfer of nutrients into foods, especially fruits and vegetables, we asked ourselves, can we push the boundaries here and infuse high levels of micronutrients into foods such that it would be food fortification or enrichment? And if this can be done, then what are the factors that would influence this process? So the first part of my study was aimed towards evaluating the effect of high pressure processing into calcium for calcium infusion in baby carrots. We chose calcium because unfortunately it's a nutrient of public health concern and a lot of US population is deficient in it. And baby carrots happen to be the most convenient and healthy snack options that a lot of US population likes. So what we did was we took our baby carrots, we put it into a calcium solution, vacuum packed it, and processed it under high pressure processing. And what we did was we varied pressure and time and the calcium solution concentration at 3 different levels, and we performed a box Pumpkin design of experiments that allowed us to perform only 15 experiments and yet be able to build a response surface. So we evaluated the amount of calcium that was infused, the amount of beta carotene that could be extracted, the texture and color. I will be focusing on texture and color. You can ask me questions about that later, but I'll focus on the calcium and beta carotene. So you'll see that the amount of calcium in baby carrots was about 20 milligrams per serving, but high pressure could infuse up to 134 milligrams per serving, which was more than 10% of the daily requirement of calcium. As we increase the calcium solution concentration, the amount of calcium infused increased. As we increase the pressure, the infusion increased. So together, increasing pressure and calcium solution concentration led to higher infusion. For beta carotene, amount of extractability in high pressure processed baby carrots was actually 5 times more as compared to raw baby carrots, which can possibly translate into higher bioaccessibility of beta carotene. So from the first part of my research, we showed that we could use high pressure processing to infuse more than 10% of daily requirement of calcium in baby carrots. So naturally, the next question we asked was, can this same finding be extrapolated to other food materials? And how would the food microstructure affect? So we did the same experiments where we infused calcium in baby carrots, celery, and mango at 5 different pressure levels in the same processing conditions. And as you can see right off the bat, that for all the 3 matrices of baby carrot, celery, and mango, the amount of infusion was significantly higher under pressure. However, within the 3 different foods, it was significantly different. So there are multiple factors that are at play here, but what I want to do is take a pause and look at how currently we understand the infusion occurs under high pressure. So what we're looking at is a very simple schematic. On the left, there is variation of pressure under the high pressure as we go throughout the processing. And on the right is a single cell of a food matrix, in this case, let's assume baby carrots. And it is surrounded by high concentration of the infused molecules, which would be calcium. So as we are increasing the pressure under high pressure, the cell wall of the food matrix permeabilizes. That means it breaks, and that reduces the resistance. So when you're holding the pressure, now the infused molecules can move in and out of the matrix. So when we release the pressure, now we end up with a cell that is enriched in the amount of infusion. So it is important that we are able to quantify the cell breakage. So what we do is we pass high electrical current that goes over a range of frequencies through these food matrices and measure the level of cell breakage, which is also quantified as a cell permeabilization index, which is ZP. So a lot of researchers hypothesize that the cell permeabilization index is the major reason why the infusion under high pressure is enhanced. But if that was the case, you will see that if we ignore the food matrix, there is no correlation between the ZP and the amount of calcium infused. In fact, if you look here, the amount of calcium infused for the same level of ZP in baby carrots and mango is significantly different, which tells us there is more work here that we need to understand. So we look at the microstructure of these vegetables. Now, given a texture or given a microstructure image like this, what we do is we draw hundreds of lines across and we obtain a graph of intensities of the pixels across the image. And this helps us quantify the cell diameter and the cell wall thickness. So if you look at baby carrots, we observed the image at 5 different points. And we looked at the texture, the microstructure. And you'll see that the cell size varies immensely throughout the texture, throughout the slice. At the very center, you see these transport tubes, which are responsible to transport nutrients. And you notice that because of the transport tubes, the microstructure is very different. The cell wall thickness is not as different between the different images, but the cell diameter goes down towards the middle of the baby carrot and then goes back up. Similar observations are for celery, where there are transport tubes around the edge, which are bigger, but the cells around it are smaller, and then they go back, their size increases again. So the transport tubes cause changes, and we'll see that the cell wall thickness is similar, but the cell diameter vastly varies. For mango, because there are no transport tubes, you'll see that there is no variation. So now the problem is, given the complexities of transport tubes in baby carrots and celery, we wanted to see which number to use. So what we did is we tagged the calcium and looked at the image of calcium infused after high pressure. So all of these bright points show you where the calcium is. So you see that the calcium is concentrated around the circle in baby carrots, in celery it's around the transport tubes, and in mango it's all over the tissue. So what we did is we looked at these specific points where the calcium was and used these images to quantify the microstructure. So the numbers we got were these. Now, if you look at the amount of calcium that was infused for these food matrices at these different cell diameters, you'll see that there's a strong correlation. Increasing cell diameter led to increased infusion. So it is very important to know that this is a correlation and not the only cause. So the takeaway here is that cell permeabilization or cell breakage is not enough to explain high pressure infusion, but what is important is the microstructure and how the cell size differs. We showed that high cell diameter correlated to higher infusion, and we developed a methodology to quantify the microstructure of fruits and vegetables. So now what is important is to understand that this is not the end of the story, this is just the beginning. What we need to do is understand how the application of pressure changes with the presence of transport tubes. So what my research now does, and you'll see this publication out soon, where we look at mathematically modeling the amount of calcium that can be infused with increasing pressure and change in microstructure. What else the scientific community can do? We need to look at how the calcium molecule actually travels throughout the matrix as the high pressure is processing. And we need to evaluate how the bioaccessibility of the infused calcium translates, because higher infusion doesn't mean higher bioaccessibility. And we need to extend this application into different food materials with different micronutrients. So the conclusion here is that we were able to show high pressure could be used as a food fortification technique to infuse significant levels of calcium. We showed a correlation between increasing calcium solution concentration and pressure led to higher infusion. We showed that microstructure is a significant variable in influencing calcium infusion under high pressure. So as any other project, this project is supported by multiple individuals and was possible by support of many different institutions. Massive thanks to the Hyperbarics Organizing Committee for letting me present my research here. Thank you for the time, and I'm happy to answer any questions you have.
Thank you for the nice presentation. It was very clear. I do have a couple of questions for you. The first one is, what were your main challenges exploring this novel application of HPP?
The main challenge was actually, there were many challenges. One of the main challenges was actually deciding what processing conditions we need to use and how can we get maximum information out of the experiments while keeping our experiments to a minimum. So deciding which kind of design of experiments we want to do was a big challenge. And later on, it was how do we quantify this microstructure that required collaboration between like 3 departments to figure that methodology out.
Okay, very nice. And the next one is, how would this application be implemented in the industry? I mean, would this be a pre-treatment followed by a step for extending the shelf life, or it be like a 2-in-1 process? How do you see it?
For me personally, I see it as just a single process. I do not see it as a pre-treatment. If we have enough studying and validation, the way I see it being implemented is if fruits and vegetables could be packaged in minimal amount of a nutrient solution and the final package goes through a high pressure process, So there is infusion that occurs, but at the same time, you're not exposing it. If you do it as a pre-treatment, you're exposing the package again. But here you wouldn't, and then that would still preserve the shelf life. And we actually did some shelf life studies, and we were able to get some good shelf life data. So hopefully this is scalable.
Okay, yeah, that's 2-in-1. It's a perfect solution.
Mm-hmm.
And that's all. Thank you for your participation and good luck for the awards.
Thank you.
And well, this is the last presentation in the category of the human and pet nutrition. Don't forget to check out the winners ceremony on the 23rd of June to find out who are the winners of the HPP Research Awards. Thank you all for watching and well, goodbye.